This is Eric Michael Lloyd, a master's in clinical psychology. Studied engineering in undergrad and took a class and was considering completing this master's in usability engineering. Engineering is still one of our favorite things to do. This paper is by Elon Musk. It's Neuralink. This one's called An Integrated Brain-Machine Interface Platform with Thousands of Channels. It's from the Journal of Medical Internet Research. The author is Elon Musk. Corresponding authors, Elon Musk, Neuralink, San Francisco, California, United States. Abstract. Brain-machine interfaces hold promise for the restoration of sensory and motor function and the treatment of neurological disorders, but clinical brain-machine interfaces have not yet been widely adopted, in part because modest channel counts have limited their potential. In this white paper, we describe Neuralink's first steps towards a scalable, high-bandwidth brain-machine interface system. We have built arrays of small and flexible electrode threads with as many as 3,072 electrodes per array distributed across 96 threads. We have also built a neurosurgical robot capable of inserting 6 threads, 192 electrodes per minute. Each thread can be individually inserted into the brain with micron precision for avoidance of surface vasculature and targeting specific brain regions. This electrode array is packed into a small implantable device that contains custom chips for low power onboard amplification and digitization. The package for 3,072 channels occupies less than 23 times 18.5 times 2 millimeters cubed. A single USB-C cable provides full bandwidth data streaming from the device, recording from all channels simultaneously. This system has achieved a spiking yield up to 70% in chronically implanted electrodes. Neuralink's approach to the brain-machine interface has unprecedented packaging density and scalability in a clinically relevant package. Journal of Medical Research, 2019. That might be volume 21, edition 21, volume 10, E16194. DOI 10.2196/16194. Introduction. Brain-machine interfaces have the potential to help people with a wide range of clinical disorders. For example, researchers have demonstrated we have developed a custom process to fabricate many neural probes, displacive neural probes that employ a variety of biocapable materials. The main substrate of dielectric used in these probes is polyimide, which encapsulates a gold thin film trace. Each thin film array is composed of a thread area that features electrode contacts and traces a sensor area where the thin film interfaces with custom chips that enable signal amplification and acquisition. A wafer-level microfabrication process enables high throughput manufacturing of these devices. 10 thin film devices are patterned on a wafer, each with 3,072 electrode contacts. Each array has 48 or 96 threads, each of which contains 32 independent electrodes. Integrated chips are bonded to the contacts on the sensor area of the thin film using a flip chip bonding process. One goal of this approach is to maintain a small thread cross-sectional area to minimize tissue displacement in the brain. To achieve this, while keeping the channel count high, steeper lithography and other microfabrication techniques are used to form the metal film at submicron resolution. We have designed and manufactured over 20 different thread and electrode types into our arrays. Two example designs are shown in Figure 1A and B. Probes are designed either with the reference electrodes on separate threads or on the same threads as the recording electrodes, referred to as on-probe references. We have fabricated threads ranging from 5 micrometers to 50 micrometers in width that incorporate recording sites of several geometries, Figure 1. I wrote on here, micron is the size of a neuron, actually. Thread thickness is nominally 4 to 6 micrometers, which includes up to 3 layers of insulation and 2 layers of conductor. Typical thread length is approximately 20 millimeters. To manage these long, thin threads prior to insertion, Perylene C is deposited into the threads to form a film on which the threads remain attached until the surgical robot pulls them off. Each thread ends in a 16 times 50 micrometer squared loop to accommodate needle threading. Since the individual gold electrode sites have small geometric surface areas, Figure 1C, we use the surface modifications to lower the impedance for electrophysiology and increase the effective charge carrying capacity of the interface, Figure 1D. Two such treatments that we have used are the electrically conductive polymer polyethylene dioxythiophene doped with polystyrene sulfonate, PSS, and iridium oxide. In benchtop testing, we have achieved impedances of 36.97, standard deviation is 4.68 kilo omega, N is equal to 257 electrodes and 257 electrodes and 56.46, standard deviation 7.10 kilo omega, N is equal to 588 for PSS, and iridium oxide, respectively. The lower impedance of PSS is promising. However, the long-term stability and bioavailability of PSS are less well established than those for iridium oxide. These techniques and processes can be improved and further extended to other types of conducive electrode materials and coatings. Just another picture of C and D. To keep the electronics package small, a novel alignment and flip chip bonding process was developed. Multi-level gold stud bumps are placed throughout the printed circuit board (PCB) to act as alignment guides and temporary holders for the thin film. A custom shuttle was used to handle, align, and place the thin film on the PCB such that holes in the thin film slide around the stud bumps, such that holes in the thin film slide around the stud bumps. The thin film is secured into place by applying force to the gold stud bumps, which flattens them into rivets. Next, the integrated chips are bonded directly to both contacts on the sensor area of the film and pads on the PCB by using standard flip coin bonding processes. A custom silicone shuttle is used to vacuum pickup rows of 40 to 50 capacitors and bond a total of 192 capacitors onto the PCB. This alignment bonding process was key to creating a package containing 3,072 channels in a 23 by 18.5 millimeter squared footprint. Robot. The thin film polymers have previously been used for electric probes, but their low bending stiffness complicates insertions. Neuralink has developed a robotic insertion approach for inserting flexible probes, allowing rapid and reliable insertion of large numbers of polymer probes targeted to avoid vasculature and record from dispersed brain regions. The robot's insertion head is mounted on a globally accurate 400 x 400 x 150 millimeter travel, 10 micrometer 3-axis stage and holds a small, quick swappable needle pincher assembly. Figures 2 and 3A. The needle, for that. The relative sizes to scale. Well, not to scale, but relative sizes. The needle is milled from 40 micrometer diameter tungsten rhenium wire stock electrochemically etched to 24 micrometer diameter along the inserted length, figure 2A. The tip of the needle is designed both to hook onto insertion loops for transporting and inserting individual threads, and to penetrate the meninges and brain tissue. Meninges, the part in between the brain and the skull. Dura mater, rachnoid, and pia mater. The needle is driven by a linear motor, allowing variable insertion speeds and rapid retraction acceleration up to 30,000 millimeters per second squared to encourage separation of the probe from the needle. The pincher is a 50 micrometer tungsten wire bent at the tip and driven both axially and rotationally. Figure 2B. It serves as a support for probes during transport and as a guide to ensure that threads are inserted along the needle path. Figure 3 shows a sequence of photographs of the insertion process into an Aragos brain proxy. This is Figure 2, the needle pincher cartridge compared with a penny for scale. A is the needle, B is the pincher, and C is the cartridge. Figure 3, insertion process into an Aragos brain proxy. 1. The inserter approaches the brain proxy with a thread, a high needle, and cannula. 2. Previously inserted thread. 2. Inserter touches down on the brain proxy surface. 3. Needle penetrates tissue proxy, advancing the thread to the desired depth. Inserting thread. 4. Inserter pulls away, leaving the thread behind the tissue proxy. Inserted thread is 4. Number 4. The inserter head also holds an imaging stack, figure 4E-G, used for guiding the needle into the thread loop, insertion targeting, live insertion viewing, and insertion verification. In addition, the inserter head contains 6 independent light modules, each capable of independently illuminating with 405 nanometers, 525 nanometers, and 650 nanometers or white light. Figure 4C. 405 nanometer illumination excites fluorescent from polyamide and allows the optical stack and computer vision to reliably localize the 16 times 50 micrometer squared thread loop and execute sub-micron visual servoing to guide, while illuminating by 650 nanometer light the needle through it. Stereoscopic cameras, software-based monocular extended depth of field calculations, and illumination with 525 nanometer light allow for precision estimation of the location of the cortical surface. There's 2 more pictures here. Figure 4, the robotic electrode inserter. Enlarged view of the inserter head shown in the inset. A. Loaded needle pincher cartridge. B. Low force contact brain position sensor. C. Light modules with multiple independent wavelengths. D. Needle motor. E. One of the 4 cameras focused on the needle during insertion. F. Camera with wide angle view of the surgical field. G. Stereoscopic cameras. The robot registers insertion sites to a common coordinate frame with landmarks on the skull, which, when combined with depth tracking, enables precise targeting of anatomically defined brain structures. An integrated custom software suite allows pre-selection of all insertion sites, enabling planning of insertion paths optimized to minimize tangling and strain on the threads. The planning feature highlights the ability to avoid vasculature during insertions, one of the key advantages of inserting electrodes individually. This is particularly important since damage to the blood-brain barrier is thought to play a key role in the brain's inflammatory response to foreign objects. The robot features an auto-insertion mode, which can insert up to 6 threads. Six threads, 192 electrodes per minute. Although the entire insertion procedure can be automated, the surgeon retains full control and, if desired, can make manual micro adjustments to the thread position before each insertion into the cortex. The neurosurgical robot is compatible with sterile shrouding and has features to facilitate successful and rapid insertions such as automatic sterile ultrasonic cleansing of the needle. The needle pincher cartridge, figure 2C, is the portion of the inserter head that makes direct contact with brain tissue and is consumable that can be replaced mid-surgery in under a minute. With this system, we have demonstrated an average of 87.1%, standard deviation 12.6% insertion success rate over 19 surgeries. In this study, precise manual adjustments were made to avoid microvasculature on the cortical surface, slowing total insertion time from the fastest possible time. Even with these adjustments, the total insertion time for this study averaged approximately 45 minutes for an approximate insertion rate of 29.6 electrodes per minute. Insertions were made in a 4 times 7 millimeter squared bilateral craniotomy with over 300 micrometers spacing between threads to maximize cortical coverage. This demonstrates that robotic insertion of thin polymer electrodes is an efficient and scalable approach for recording from large numbers of neurons in anatomically defined brain regions. Pretty cool picture of it. This machine here that the surgeon uses to implant the electrodes. Neuralink. Figure 5. A packaged sensor device. Individual neural processing application-specific integrated circuit capable of processing 256 channels of data. This particular packaged device contains 12 of these chips for a total of 3,072 channels. Polymer threads on a Perylene C substrate, titanium enclosure, lid removed, digital USB-C connector for power and data. Electronics. Chronic recording from thousands of electrode sites presents significant electronics and packaging challenges. The density of recording channels necessitates placing the signal amplification and digitization stack within the array assembly. Otherwise, the cable and connector requirements would be prohibitive. This recording stack must amplify small neural circuits, lesser than 10 microvolts RMS, while rejecting out-of-band noise, sample and digitize the amplified signals, and stream out results for real-time processing, all using minimal power and size. The electronics are built around our custom Neuralink application-specific integrated circuit, ASIC, which consists of 256 individually programmable amplifiers, analog pixels, on-chip analog to digital converters, ADCs, and peripheral control circuitry for serializing the digitized outputs. The analog pixel is highly configurable. The gains and filter properties can be calibrated to account for variability in signal quality due to process variations and the electrophysiological environment. The on-chip ADC samples at 19.3 kilohertz with 10-bit resolution. Each analog pixel consumes 5.2 microwatts, and the whole ASIC consumes approximately 6 microwatts, including the clock drivers. Performance of the Neuralink ASIC is summarized in Table 1, and a photograph of the fabricated device is shown in Figure 6A. The Neuralink ASIC forms the core of a modular recording platform that allows for easy replacement of constitutive parts for research and development purposes. Figure 6. In the systems discussed here, a number of ASICs are integrated into a standard PCB using flip chip integration. Each system consists of a field programmable gate array, real-time temperature, accelerometer, and magnetometer sensors, and a single USB-C connector for full bandwidth data transfer. The systems are packed in titanium cases that are coated with perylene C, which serves as a moisture barrier to prevent fluid ingress and prolong functional lifetime. We describe two such configurations that we have built, a 1536 channel recording system, System A, and a 3072 channel recording system, System B, summarized in Table 2. System A employs the current generation Neuralink ASIC, while System B uses an earlier revision with comparable functionality but poorer performance specifications. And it has a table here that has variables and values that correspond with those. Number of channels, 256. The gain, the decibel level, that would be lowercase d, uppercase b, 42.9 through 59.4. Bandwidth in kilohertz is 3 through 27 kilohertz. Input reference noise, 3 hertz through 10 kilohertz. And I think that's microvolts. RMS is the subscript. 5.9. Maximum differential input range. Microvolts PP. 7.2. Analog to digital converter resolution. Bit is 10. The value for that. Analog pixel power, microwatts, 5.2. That's Table 1, Neuralink Application Specific Integrated Circuit. There's a picture here of a Figure 6, a picture of the thread implantation and packaging. An example perioperative image showing the cortical surface with implanted threads and minimal bleeding. B, package sensor device, System B, chronically implanted in a rat. So it actually has that device implanted in a rat. And the USB-C port as well with the 3,072 channels. Check it out. There's another table here. Table 2, two recording system configurations. It has the values here, the number of channels, the sampling rate in kilohertz, total system power consumption in microwatts, and total system size. That would be millimeters cubed, I think, or mm3. Implant weight, grams. So system A, 1536, 1,536 channels. System B, 3,072. Sampling rate in kilohertz is 19.3 for System A and 18.6 in System B. Total system power consumption in microwatts is 550 for System A, and for System B it's 750. Total system size in millimeters cubed is 24.5 times 20 times 1.65. And for System B, it's 23 times 18.5 times 2 millimeters. Implant weight in grams. System A weighs 11 grams and System B weighs 15 grams. System B was designed to maximize channel density and is used for applications that demand extremely high channel count. In contrast, System A was designed to facilitate faster and more reliable manufacturing. It can be built 5 times faster than System B with better yields. And it's lighter. An Ethernet-connected base station converts the data streams from these systems into multicast 10 gigabyte Ethernet user datagram protocol packets, allowing downstream users to process the data in a variety of ways. For example, visualizing the data in real time, or writing the data to disk. Each base station can connect up to 3 implants simultaneously. These devices are further supported by a software ecosystem that allows for plug-and-play usability with zero configuration. Neural data begins streaming automatically when a cable is connected. Wow. You just connect the cable to the USB there, and data just begins to stream. Wow. Neural data, that is brain, data from the brain. Electrophysiology. We have implanted both systems A and B in male Long Evans rats, as described in the section Robot. All animal procedures were performed in accordance with the National Research Council's Guide for the Care and Use of Laboratory Animals, ethics, again, and were approved by the Neuralink Institutional Animal Care and Use Committee. Okay. Electrophysiological recordings were taken as the animals freely explored an area equipped with a commutated cable that permitted unrestricted movement. System A can record 1,344 of 1,536 channels simultaneously. The exact channel configuration can be arbitrarily specified at the time of recording. System B can record from all 3,072 channels simultaneously. Digitized broadband signals were processed in real time to identify action potentials, spikes, using an online detection algorithm. Spike detection requirements for real-time brain-machine interface are different from most conventional neurophysiology requirements. While most Electrophysiologists spike sort data offline and spend significant effort to reject false positive spike events. Brain-machine interface events must be detected in real time, and spike detection parameters must maximize decoding efficacy. Using our custom online spike detection software, we found that a permissive filter that allows an estimated false positive rate of approximately 0.2 Hz performs better than setting stringent thresholds that may reject real spikes. Data not shown. Given these considerations, we set a threshold of greater than 0.35 Hz to quantify the number of electrodes that recorded spiking units. Since we typically do not sort our data, we do not report multiple units per channel. Brain-machine interface decoders commonly operate without spike sorting with minimal loss of performance. Moreover, recent results show that spike sorting is not necessary to accurately estimate neural population dynamics. Data from a recent experiment using System A are shown in Figure 7 and 8. In this experiment, 40 of 49 attempted insertions were successful. That's 90% for a total of 1,280 implanted electrodes, of which 1,020 were recorded simultaneously. The broadband signals recorded from a representative thread show both local field and spiking activity, Figure 7. A sample output of the spike detection pipeline is shown in raster form in Figure 8. In this example, two overlapping recording configurations were used to record from all 1,280 implanted channels. On this array, our spiking yield was 43.4% of the channels, with many spikes appearing on multiple neighboring channels, as has been observed in other high-density probes. On other System A arrays, we observed a spiking yield of 45.60%. And this is Figure 7. The broadband signals recorded from a representative thread. On the left side, you have broadband neural signals that are unfiltered. Wow, these are actually ray recordings. That's so cool. Simultaneously acquired from a single thread, 32 channels, implanted in a rat cerebral cortex. Each channel row corresponds to an electrode site on the thread. Schematic at left, sites spaced 50 micrometers. Spikes and local field potentials are readily apparent. Right, putative waveforms unsorted. Numbers indicate channel location on thread. Mean waveform is shown in black. Check that out. So cool. And this is the discussion part with a really big picture. It takes up about 80% of the page of this Figure 8. Figure 8. Our devices allow the recording of widespread neural activity distributed across multiple brain regions and cortical layers. Left, thread insertion sites, colored circles, are indicated on the rendered rodent brain. On the right, raster of 1,020 simultaneously recorded channels, sorted per thread. Color corresponds to the insertion site. Inset and large raster of spikes from a single thread. This thread corresponds to the one shown in Figure 7. it says color coded, but this is, and still is in black and white. That little subsection right there in that box shows 200 milliseconds of time. And this section is 4 millimeters of the lab rat's brain. 4 millimeters right here. Discussion. We have described a brain-machine interface with a high channel count and single spike resolution. It is based on flexible polymer probes, a robotic insertion system, and custom low-power electronics. This system serves two main purposes. It is a research platform for use in rodents and serves as a prototype for future human clinical implants. The ability to quickly iterate designs and testing in rodents allows for the rapid refinement of devices, manufacturing processes, and software. Because it is a research platform, the system uses a wired connection to maximize the bandwidth for raw data streaming. This is important for performance assessments and crucial for the development of signal processing and decoding algorithms. In contrast, the clinical devices that derive from this platform will be fully implantable, which requires hermetic packaging, and have onboard signal compression, reduced power consumption, wireless power transmission, and data telemetry through the skin without percutaneous leads. Modulating neural activity will be an important part of next-generation clinical brain-machine interfaces. For example, to provide a sense of touch or proprioception to neural prosthetic movement control. Wow. Therefore, we designed the Neuralink ASIC to be capable of electrical stimulation on every channel. So I don't know if I'm missing a page or not. Every channel, yeah. And then there's a video. I can't play the video. It's just a paper copy of the article. But the video, this is Multimedia Appendix 2, and it has the references here as well. You can go ahead and check that out. I put the DOI as well. I read out the DOI as well, so anyone could check that out. Has all his references and everything there as well. Pretty cool stuff. Check it out. Usability, engineering, say biomedical engineering at its finest. Thank you.